हम्म? सर, इसमें मैं स्टार्ट स्ट्रीमिंग कर दिया, बट लिंक कहाँ से मैं शेयर? यूट्यूब कनेक्ट हो गया उसी लिंक से? अरे ये तो गड़बड़ हो जाएगा सर। हम्म? नहीं। यूट्यूब डैशबोर्ड ना? डालूं? ओबीएस ना? ऑलरेडी शेयर कर रखा है किसी ने। किसी ने ऑलरेडी शेयर कर रखा है। Good evening, participants. I hope you are doing well in a lockdown period or corona pandemic. My voice is audible to all. Please comment. Someone are saying my voice is not audible. Okay, no problem. Good. सेम वीडियो तो नहीं बनेगी, डिफरेंट होगी ना? Good evening, good evening, good evening to all. Now my voice is clear to all. Is it okay? Okay, good. We will wait for 2 or 3 minutes so that others people can join. In the today's lecture or in a session, we will discuss about the review, okay, uh, for your internship program, right? So, how to write a review paper, I will tell you, okay? So, I will share with you some topics on a recent uh, research, okay? So you have to write review paper. I will discuss how you will write review paper, okay? First, because your internship will start from today, and you have enough time to write a review paper. My voice is audible to all or not? Is it very much clear or weak? Please comment. Someone are saying that my voice is not clear. Please comment. But someone are saying that my voice is as good as possible. We can sit for Google Meet. Okay. Uh, we will try, but 2 3 lectures I will take on the YouTube. After that, we can shift on Zoom or Google Meet. Okay. Because some issues are there, uh, that's why we cannot shift on Zoom or Google Meet. Okay. Someone are saying that my voice is very much clear. Okay. I am sharing link again on Telegram. Please join. Before starting this session, we will discuss about the topics on which you have to prepare a review. Okay. I will share the list with you. So first topic is role of bioinformatics in biology. Okay. Now second thing is genome editing. Now third thing, drug designing, and next one is genetically modified food. Next one is genetically modified food synthesis. Next is characterization of genetically modified foods. How you will characterize, what are the methods to characterize genetically modified foods. Now next one is ethical issues of genetically modified foods. Okay. Next one is future of genetically modified foods. Okay. Now last one is gene therapy. So you have to choose one topic from this list. Okay. If you have any doubt, you can ask. Okay. Now I will give the brief introduction of all these topics. What are the meaning of these topics? First is the role of bioinformatics in biology. So you have to discuss the role of bioinformatics. Without bioinformatics, you cannot go for anything in a biological system. Okay. If you are doing molecular biology, so you have to design primers, you have to construct vector. Everywhere you will use bioinformatics. You will go for the sequencing, you will analyze your data, you will go for the bioinformatics. Okay. You are isolating a protein, you have sequenced a protein, then you will analyze that protein on the basis of homology modeling, and your amino acid sequence will match with which protein, that will be your protein. Okay. Otherwise you will go for the X-ray, crystallography, or NMR, nuclear magnetic resonance, right? So you have to write about the role of bioinformatics in biology. Is it clear? Is it clear? Okay. I will increase the font size. Now it will be very clear. Okay, I will explain this again, no problem, if someone are demanding this thing. Okay, first I am going to discuss first point again, role of bioinformatics in biology, right? Then you have to discuss the role of bioinformatics, where bioinformatics required in a biological system. Without bioinformatics, you cannot do anything in a research. Okay, because you are if you are doing molecular biology research, then you have to isolate DNA and you will go for the sequencing. Then you will analyze your data. Okay, then you will require bioinformatics tools for the analysis of sequencing data. If you are going for the cloning, you require vector or primer. You will use different software for designing of primers and construction of vector. Okay, so everywhere, if you are going for the evolutionary biology, you will go for the phylogenetic tree preparation. Okay, so everywhere you require bioinformatics. Without bioinformatics, it is not possible. Okay, so you know, in a human, more than 3 billion base pair DNA is present. So Excel sheet is not enough to compile all the data of nucleic acid or genomic DNA of human. That's why we required bioinformatics. So from the before you will collect all the research paper from present to last 5 to 6 years, and you will read all these papers. You will find out the what type of research were performed and how bioinformatics play a important role in biological science. Okay, and how you will write, I will discuss later this thing. First I am discussing about the important topics on which you have to write a review paper. Okay, yes, this lecture is recording in a YouTube, so you can watch it again whenever you need it. Now I am going to discuss the second topic that is also very much important, because this is the latest topic and recent lots of research are going on in a genome editing. Okay, before some time we were going for the genetic engineering, so we can change one or two gene through a genetic engineering, but with the help of genome editing techniques, we can target more than 1 2 3 4 5 6 genes at a single time. So some latest tool like CRISPR Cas9, you have to discuss about the CRISPR Cas9 ZFN zinc finger nucleases. You may note down, take your notebook and pen, so that important points you can note down. Okay, first you take notebook and pen. So in a genome editing, the latest tool is CRISPR Cas9, through which we can target multiple genes. You can mutate multiple genes. Okay, for the production of transgenic plant, transgenic animals, transgenic microorganisms, we are using latest technology of genome editing, that is CRISPR Cas9, that is very much popular in a recent time. Okay, so you have to focus on a CRISPR Cas9, role of CRISPR Cas9 in a research of biological science. Okay, so you have to know how it works. Okay, you have to know the mechanism of CRISPR Cas9. base, and you have to also know the basic mechanism of ZFN, zinc finger nuclease, right? This is the old tool for genome editing. Now we are not using uh, in a genome editing because the latest tool is available, genome editing, CRISPR-Cas9, right? Now we are going on a next point, drug designing. Okay, this is a topic of bioinformatics. Uh, so how you will design a drug, it will work at a specific target. So docking is required here for the designing of drug. What should be the structure of any drug? Okay, so lots of research has been done. This is a basic topic of pharmaceutical science. Okay, so the participants who have from pharmaceutical background, they can choose that topic. Okay, this topic will be good. Now most important thing is here, genetically modified food. Everyone is aware about the genetically modified food because food scarcity is there. Our population is increasing day by day, but food production is not increasing regularly. Okay, that's why we have to produce a transgenic plant for the production of high production level. Okay, and lots of genetically modified foods have been prepared using genetic engineering or other techniques. Okay, so you have to search uh, the research paper on genetically modified food. Okay, now next thing is genetically modified food synthesis. How you will synthesize a genetically modified food? What should be the different from the wild type of genetically modified food? Okay, my voice is clear. Someone is saying that my voice is very clear, but you can check your connection or earphone, maybe problem in your earphone or network problem. Okay, please check. Okay, I will repeat it again. GM food synthesis. So you have to discuss here about the genetically modified food synthesis. How you will prepare genetically modified food. How? What is the basic difference between wild type of food or genetically modified food, and how you will synthesize it? Okay. Now, okay, someone is saying it's low in phone, but it's clear in laptop. Okay, you use laptop. Okay, from next day you try to connect in a laptop. It will be good because I will show you some uh, website like NCBI. We will search nucleic acid sequence, then we will go for the blast, and we will perform different types of analysis. So it will be good because all these things are not possible in a mobile phone. So you try to connect in a laptop, it will be good. Okay, Now, next topic is characterization of genetically modified food. Okay, how you will characterize a genetically modified food? Okay, different techniques are there, so you have to discuss uh, different techniques, and all these techniques uh, have been used for which type of genetically modified food. Okay, now main important thing is ethical issues of genetically modified food. Okay, uh, you have to discuss about this, because genetically modified may be harmful for the human life or environment or other in a other way. So you have to discuss here the ethical issues. It is good or bad. You have to discuss here both issues, positive and negative. How it is bad, how it is good, how it is safe. We have to use or not. Okay. Yes. Uh, everyone have to write. Uh, just a wait. A minute. I enrolled for training program. I also have to write review paper. Yes, you have to write a review paper. Okay, you will learn new thing. This will be beneficial for your career. Okay. Now, next point is future of genetically modified food. In a future, what will be the scope of genetically modified food? You have to discuss this thing. You have to develop own approach after reading so much papers. Now, last topic is gene therapy. Okay, you have to discuss about the gene therapy here. Uh, what is gene therapy? Anybody knows this thing? Gene therapy. This is a very common topic. I expect from you, you know very well this thing. Yes, before end of this course, you have to submit this review paper. Okay, good. Most of the people knows about the gene therapy. If someone do not know, then I am telling you about the gene therapy. Gene therapy. Uh, some diseases are due to gene, faulty of genes. Okay, maybe that protein may be faulty or may cause some type of disease. So we can replace that gene. Maybe some diseases like inherited disease. So if we will change a specific gene, then we can cure that specific disease. Okay, so you have to write, this is a broad topic, you may choose. Okay, so these were the topics, you may choose any topic. Okay, to write review paper. Now I am going to discuss about how to write a review paper. Okay, so first of all, you have to write a abstract. Okay, in an abstract you have to show the brief idea. Brief idea means a conclusion type, like conclusion type means whatever you have read in all the papers and you are going to discuss in your paper, that should be summarized in a 200 to 300 words. Okay, is it clear to all? Is it clear to all? Please reply first. Okay, I will tell you. Uh, Someone are saying that I never written any paper, so I will tell you how to write a paper. How you will search research paper. Uh, okay, first we will discuss how to write. Okay, then I will tell you how you will search research paper. Okay, one by one. We will finish one thing, and then we will start the second thing. Okay, now this is the review paper format, and I will share with you. First will be a title page. uh, in the title page you have to write your name correctly, and email id, and your college name or university name, and please wait for 1 minute.